Okay, greetings everyone, and welcome back for the final session of our summit today, which I think promises to be a really exciting and interesting one, covering some of the most innovative and cutting edge areas in the field. We've got three speakers, a couple by video, one live, and all available for Q&A after the session. So I'll kick things off. Our first speaker is Associate Professor Peter Johansson. Peter's a Senior Research Officer in the Oncogenomics Group at QIMR Berghofer in Brisbane. He's one of our country's leading computational biologists in the cancer space. He's got decades of experience in working to develop computational methods to help us interpret large data sets, previous experience in the machine learning field, and moved really deeply into the cancer genomic space in the late 2000s via a stint at NCI in Bethesda. He was recruited back to Australia in 2012, where he joined the Oncogenomics Group, and it's certainly great to have him in our national community. So I'll throw to you, Peter. Thanks very much for your talk.
I'm grateful to the organizers for inviting me to speak here today. I want to talk about two of the most prevalent high to medium risk genes in uveal melanoma, namely BAP1 and MBD4, which are both located on chromosome 3. Mutations in BAP1 and MBD4 occur both in the germline, increasing the risk for and they also occur somatically in the tumor. I will talk a bit about a simulation study we did of surveillance of BAP1 mutation carriers and show some data about the risk that you get from MBD4 mutations, and that patients with these mutations might be good candidates for immunotherapy. This is a cohort of 103 patients. I want you to focus on the first row, where purple and black indicate mutation of BAP1. And as you can see, about 1/3 of the samples have a mutation in BAP1. Importantly, almost all of them have monosomy 3, which means that the other copy of BAP1 is lost as well. Most of the mutations are somatic, but there are 2 cases of germinal mutations marked by a white blob in the black rectangle, and those are the 2 I want to focus on today. So germinal mutations in BAP1 leads to BAP1 tumor predisposition syndrome, leading to a wide range of different cancers. There's a core spectrum, including 4 cancers. Uveal melanoma is the most common with 25%, and it also includes mesocutaneous melanoma and RCC. And then there is a long tail of various cancers that occur less frequently. Suggestions for long-term surveillance of carriers of BAP1 mutations have been published by Mohammed Abdel Rahman in Clinical Genetics. And it includes eye and skin checks for melanoma, and for meso and RCC, the checkups are more intrusive and therefore more debated. We wanted to evaluate such surveillance, and to do that we used simulations with a computer model. And this is a simple sketch of the model where every person starts in the healthy state, indicated by the yellow circle here, and in each time step a person either stays healthy or dies completely unrelated to BAP1, or they develop one of the 4 core cancers here indicated by blue circles. And then when they have developed the cancer, they can be diagnosed either early or late. They get treatment, which leads to either that they are cured and then they're back in the yellow healthy state, or they die from the cancer. We ran this for 2 settings, with surveillance and without, and simulated it for 10,000 people and collected the stats that we are interested in. One obvious statistics that we want to look at is how old people get. And here is the age distribution. The blue curve is for the group of people without surveillance, and the red curve is for people with surveillance. And as you can see, at the early peak, at around 50 years old, the blue curve is substantially higher than the red curve, meaning that the non-surveilled group have more people dying prematurely. And on average, the surveilled group become 4.9 years older in these simulations. Similarly, for the cost, we see that the red peak is shifted right compared to the blue one, which means that when you introduce surveillance, the baseline cost bumps up a bit. Importantly, for higher costs, the red curve is below the blue curve, which means that there are fewer high-cost people in the surveilled group, which is basically because there are fewer patients with late-stage disease, and that's really where the high cost kicks in. And on average, a surveilled person costs the health system $6,200 more in these simulations. And if you translate it to, you can also say that it's $1,300 per increased years, which is the typical way to measure this, and it's much less than the threshold in these sort of studies. So now I want to change gear a bit and talk about MBD4. Germinal mutations in MBD4 was first reported back in 2018 by this French group. They had a patient that had received immunotherapy and it responded much better than the typical patient. When they sequenced the tumor, they noticed that they had much more mutations than a typical has, and most of the mutations were these sort of S to C to T transitions at CPG positions, which is exactly the kind of mutations that MBD4 is involved in repairing. So it all makes sense. Sequencing also showed that the tumor had monosomy 3, so the other copy of MBD4 was lost as well in the tumor. We could confirm this finding in our cohort, where we had a patient that also had received immunotherapy. She survived with metastatic disease for more than 24 months. And then we found another case in public data from Bill Harbour. And then in a larger cohort, we found another 2 cases. And they all showed the same pattern with monosomy 3, or 2 cases of isodisomy 3, but it's in this context the same thing. And high level of mutation burden, and all basically the same type of mutations. And now it's starting to become a pretty large number of cases here suggesting that the mutation is predisposing to uveal melanoma. The French group looked at that properly last year, where they looked at all patients coming to their clinic between 2013 and 2018 being diagnosed with primary they identified 8 patients with deleterious germline MBD4 mutation, or 0.7%, which is much higher than what we see in the healthy population, and translates to a 9-fold increased risk. And we all know that people are excited about immunotherapy, but it hasn't been a really great success in uveal melanoma. Here is a study from last year where the median overall survival was about 22 months, which was good enough to get this drug FDA approved, but compared to cutaneous melanoma, it's not good at all, because there we have a median overall survival more than 5 years. So it's a big gap between uveal melanoma and cutaneous melanoma. And one big reason for that is probably that uveal melanoma typically has fewer mutations, and that's a known marker for immunotherapy. So that's why people have been thinking that these patients with MBD4 mutations that have much more mutations should, in theory, be good candidates for immunotherapy. And just recently, this month actually, the same French group came out with a paper where they retrospectively looked at all their patients that had received immunotherapy. So in total, 300 patients with metastatic and they identified 5 people with deficient MBD4. Interestingly, only 2 of them had germline mutations, and the 3 others had a somatic mutation plus LOH. And the same pattern, that the tumors with MBD4 mutations have much more mutations, and more importantly, they responded much better to the immunotherapy. And when looking at survival, the patients with MBD4 mutations survived significantly longer, both when looking at progression-free survival and overall survival. So in summary, I've been talking about BAP1 and MBD4, both located on chromosome 3. They have mutations, both as germline mutations, predisposing to uveal melanoma, and tumors also have somatic mutations. I mentioned a simulation study suggesting that surveillance of BAP1 mutation carriers are most likely cost-effective, and I've showed that an MBD4 mutation increases the relative risk with 9-fold, and these patients are most likely good candidates for immunotherapy. And lastly, I just want to thank everyone involved in these studies. It's a lot of people, as usual. Thank you for your attention.
Okay. Thank you very much, Peter. So we're just moving on straight on to Sarah Coupland now, and we'll take questions at the end of the session. So our next speaker is Professor Sarah Coupland, and she'll be talking about proteomics in uveal melanoma. So Professor Coupland's a senior consultant histopathologist at the University of Liverpool Foundation Hospital Trusts, and she's an expert in ophthalmic or ocular pathology, and also hematopathology and molecular and digital pathology and biobanking. So she established the Liverpool Ocular Oncology Research Group in 2006, and this is a multidisciplinary team focusing on ocular melanomas, lymphomas, and other tumours. And she's the George Holt Chair of Pathology at the University of Liverpool. She holds a number of additional roles within pathology and ophthalmology, and she's published over 300 scientific articles, and she's won numerous awards. And very much looking forward to your talk, Sarah.
Thank you very much, Dan. Can you see my slides?
Yes.
And you obviously can hear me.
Yeah.
Okay. Thank you very much for inviting me to talk at this summit. I'm going to be giving you an overview of proteomics in uveal melanoma. So this is just the outline of my talk, and so I'll dive straight in. So I think we all take it for granted that there has been an omic revolution, and similar to the scale of the industrial and the digital revolutions. And as we've just heard from the previous speaker, with the incredible advances in genomics, transcriptomics, proteomics, and metabolomics, so that we can really get to understand the details of cells, not only tumor cells, but also our background germline genomic and proteomic profiles. And whereas the human genome was cracked, so to say, in the year 2000, it took longer to produce the human proteome. And this was an effort, an international effort, coordinated through the Human Proteome Project, leading to this Nature paper in 2014. And since then, we have detailed analyses of virtually every single organ in our bodies, providing us with a protein profile of these organs, so that excuse me, that we can better understand the biology of these tissues, and obviously through that the biology of disease. So in Sweden, the Human Protein Atlas was first designed, and this incorporates, it's a fantastic resource which combines all sorts of information regarding immunohistochemistry, Western blotting, and excuse me, also all of the information regarding mass spectrometry, and so this is a resource which is continually being updated and provides tremendous information for all scientists. Similarly, with respect to functional proteomics, is the Cancer Protein Atlas, which is located in MD Anderson Cancer Center, and this incorporates a large amount of material and data that was produced through the Cancer Genome Atlas, a project which spanned numerous years and involved a large number of institutes, but it was essentially run from the NIH. So when it comes to uveal melanoma, there have been a number of studies over the years using a variety of different techniques. And I'm just going to go through some of the studies to outline what has been revealed through the differing techniques, and on the basis of cell cultures, cell lines, primary tumors, and in some cases, but very limited, metastatic material. So I think the pioneer in this area was Maria Pardo, who did whole proteomics on one single human uveal melanoma cell culture. And she found approximately 270 spots, which represented over 700 proteins. And a good 90% of these were novel when it came to understanding uveal melanoma. And they could be broken down into their location within the cell, but also then broken down in terms of the function. And there are recurring themes, and one that includes heat shock proteins. So we collaborated with a Danish group, and we used archival frozen tissue with all of the associated clinical data and also genetic data. And this was together with Professor Henrik Vorum. And we found one particular biomarker, which was heat shock protein 27. And this was associated with chromosome 3 loss. We then looked at paraffin tissue, and this was a larger cohort than the original study. And we found that indeed that we could use HSP27 immunohistochemistry to look at or predict the outcome of the patient. It did correlate with chromosome 3. This was confirmed by other groups. However, this was all in at the time of 2012, and things research progresses, and as we all know, BAP1 became a prominent prognostic marker, and so we moved on from HSP27 and have, as I mentioned previously in the question session, we do regular immunohistochemistry for BAP1 rather than for HSP27 any longer. However, we did decide to continue with the proteomics, and we looked at this so-called secretome of uveal melanoma cell lines, of uveal melanoma samples from patients, and we compared these with normal choroidal melanocytes. And using a variety of different techniques, but particularly liquid chromatography, mass spectrometry, or LC-MS for short, we could differentiate the proteins which were secreted from these cultures, and we saw a difference between the so-called low and high risk uveal melanomas. This was work undertaken by Dr. Martina Angi, who was a PhD student at that time, and then it was continued by another PhD student, Sam Prendergast, who then looked at this secretome and looked to see whether these the proteins were packaged in so-called exosomes or were they free floating. And we found that the vast majority were within exosomes. And this was a whole area of research at this time, and where it was thought, and continue the hypothesis is that tumor cells secrete secretomes which pass into the blood and then or into the lymphatics, and they prepare the metastatic niche of the tumors. And so we looked in detail, and we thought that exosomes may have a role in preparing the pre-metastatic niche within the liver, and maybe interacting with the hepatic stellate cells, or indeed with hepatocytes. And so Sam looked at various uveal melanoma cell lines and could isolate these exosomes using electron microscopy. And you can see that they have sort of almost like an erythrocyte disciform shape, and they are produced by not only the uveal melanoma cell lines, but also by the primary cell cultures. He then went on and examined the content of the exosomes, these exosomes using a variety of different proteomic techniques. And one consistent feature was this hepatic stellate cell activation factor, which we believe, and others have also validated, that interacts with the hepatic stellate cells, which in turn lead to fibrosis, and this is part of the evolution of the uveal melanoma metastases. So a number of researchers were also looking at exosomes, which are also called ectosomes, just to be confusing, or extracellular vesicles. And this group looked at one cell line, others looked concentrated on the microRNAs within the exosomes, and others have also looked at the content within the eye. So we have continued our work in looking at this so-called crosstalk between the uveal melanoma cells and also those within the liver. And this is the work of a postdoc, Dr. Karen Orton, who's been doing co-culturing of uveal melanoma cell lines with hepatic stellate cells and also hepatocytes, and using a citation which enables you to video this interaction over days. You can fluorescently label the different cell types and see how they interact with each other, and then also look and observe what happens when you treat these organoids or spheroids, as they're called. This also has been an area of research by this group in New York, who looked at 3 different cell lines and compared them to normal melanocytes. They too found ectosomes or exosomes within these cell lines, and then found that if you that there was this crosstalk between the uveal melanoma cell lines and the liver cells, and also there was one particular factor called MIF, or macrophage migration inhibitory factor, and that if you knock this down, this would result in decreased migration of the uveal melanoma cells. And this is promising in terms of potential treatments in future. This factor MIF was actually first described by Jonathan Crabb, who works in Cleveland. He used an earlier technique of proteomics called iTrack, and he found this in approximately 15 samples. Interestingly, he found it in all uveal melanoma samples, and it wasn't in a particular cohort. And we've worked together with John, and we had a cohort of 100 uveal melanoma samples, and these were all primary tumors, and these were divided into low and high risk tumors. And indeed, this MIF doesn't really differentiate between tumors which are likely to metastasize and those that are not. However, he was able to, on the basis of very detailed analysis, come up with differentiating proteins which enable us to stratify the patients in terms of their risk of metastasis. And this is a comparison with normal choroid. So it's really quite distinctive. There are 32 differentiating proteins, which he's putting together as a panel, and you can see the sensitivity and specificity of this panel with in this ROC curve. This study was also quite a major finding of this study was also looking at the immune system, and he could demonstrate that there was a particular immune profile of the proteins within the metastasizing uveal melanomas. And he then compared this profile with what has been demonstrated in 3 transcriptomic studies, and this included one of ours, or actually 2 of ours, which we published in Pathology, Journal of Pathology, and also in Cancers last year, together with the single cell analysis undertaken by Bill Harbour's group. So there was consistency in the immune profile of between the studies, and it also revealed, John Crabb's study also revealed new proteins which could be targeted amongst the new immunotherapies which are in development. Just to say that simultaneously people have been looking at liquid biopsies. So these are older studies which have looked at osteopontin and MIA, and this has been regularly looked at by Jacob Pier's group in Israel. Interestingly, Maria Pardo also came up with a number of proteins in the serum of patients with uveal melanoma, which have been since validated by this group from China. Then others have looked within the aqueous humor and the vitreous, and you can see it's really quite complicated. The range of the range of samples which have been analyzed before and after brachytherapy, and a number of different proteins have been highlighted. But I think in terms of differentiating within the samples of the aqueous or the vitreous, probably panels are required, and obviously these are still within the research arena and require validation. So what's next? I think a very promising technique is spatial orientation of proteomics using tissues. This is either using formalin fixed paraffin embedded material or fresh frozen tissue, and so that you get not only analyses of the protein profiles, but in the anatomical tissues, so you can see the interaction between the cells. So this is called mass spec imaging. And I think really with all of the different techniques which are rapidly being developed and also are becoming more accessible in terms of their costs, we can have a combination of data in terms of what is being called tissue phenomics, and so that we can look at not only the genomics, the transcriptomics, the proteomics, and even the metabolomics, and combine all of these data using AI. And I think instead of perhaps concentrating on the primary uveal melanomas, we should be concentrating more on the metastases, because this is obviously the real clinical challenge at the moment. And so by concentrating on the metastases, we may be able to reveal new targets, treatment targets, which will be the future of clinical trials. So with that, I'd just like to thank you very much for your attention. I'm happy to take questions. And I'd just like to acknowledge all of our sponsors and also the patients who've raised money for our research group. Thank you very much.
Thank you very much, Sarah. Thank you. Thank you. We'll just we'll have Aaron's talk next, and then we'll stop for questions. So Aaron Beasley is an early career postdoctoral researcher at the Centre for Precision Health at Edith Cowan University in Perth, Western Australia. His research is focused on liquid biopsies in uveal melanoma, with specific focus on circulating tumour cells and circulating tumour DNA. His talk is therefore appropriately titled Liquid Biopsy in Uveal Melanoma. Thanks, Aaron.
Hi everyone, my name is Aaron. Today I'm going to be talking about liquid biopsies in uveal melanoma, and I apologize if I sound a bit nasally. I have a bit of a cold. So liquid biopsies, as the name suggests, are techniques used to assess fluids in which the tumor has contact with. So traditionally this generally refers to blood, but it really can be any fluid. So this could be anything from saliva to cerebrospinal fluid to even the humors within the eye. The most prevalent markers that are assessed in liquid biopsies are circulating tumor cells and circulating tumor DNA. So circulating tumor cells are cells that have broken away from the tumor and they disseminate, for example, into the blood, and this is how they move around the body. And circulating tumor DNA are molecules of DNA that have formed as a result of cell death or apoptosis and necrosis. So they die and are released into the blood, and you can detect them using PCR or sequencing. So liquid biopsies have been used in a range of applications. These range from diagnostics or cancer screening to prognosis, to monitoring for recurrence, to detecting mechanisms of resistance, and for treatment selection. So why would you want a liquid biopsy over, or at least in conjunction with a traditional biopsy? So I think it's important to note that the classical tumor biopsy is the gold standard. I mean, it is the ground truth of what the disease is, but there are some limitations to it. And these are primarily the invasiveness of the procedure. They only represent what you take at the time of the biopsy. So for example, if you take a fine needle or a core biopsy, you're only sampling a very small proportion of that disease. And serial biopsies are quite impractical. On the other hand, liquid biopsies are generally minimally invasive. So for example, think of a venipuncture on your forearm. You can serially monitor using a liquid biopsy. So for example, you can take a blood test every couple of weeks if you wanted. And liquid biopsies generally represent the sum of tumors present in a patient at one time. So for example, if you have metastatic disease and there's a few lesions throughout your body, all of those analytes spill into the blood. And then when you take a blood sample, you have a representative proportion of those different tumors in that one sample. So it's very good for understanding heterogeneity of disease. So because we're in Melbourne for this AOMA, I thought it would be fun to have some circulating tumor cell trivia. So circulating tumor cells were actually discovered by Dr. Thomas Ashworth from Melbourne Medical School back in 1868. So while we've known about circulating tumor cells for a long time, it's only really in the last couple of decades that clinical applicability has really been described. So in primary uveal melanoma, circulating tumor cells, basically they break away from the tumor and they move into the bloodstream. And this is how patients with high-risk disease will have the disease spread. Now, this is an example of a circulating tumor cell, and you can see this green and blue. So that's a circulating tumor cell, and you can see it's much larger than the surrounding white blood cells. So these are the blue and purple. So circulating tumor cells can be actually isolated in a few different ways, but the most prevalent ways are immunomagnetic isolation and physical property-based isolation. And in uveal melanoma, historically, there's been quite a lot of circulating tumor cell analysis. This dates back to 1969, where the first paper described using standard staining techniques and standard microscopy techniques, detection of circulating tumor cells. Then this moved into RT-PCR in the 90s, and then immunomagnetic isolation in the early 2000s, and then in the 2010s, microfluidic-based methods. Now, the field has kind of mostly settled on immunomagnetic isolation, but no matter the technique used, there is a general trend in most papers that increasing levels of circulating tumor cells are associated with poor prognostic features such as ciliary body involvement and in general poor disease-free, poor progression-free, or poor overall survival. So we recently published a multi-marker immunomagnetic isolation method, and this was designed to improve the detection of circulating tumor cells in patients with primary disease. So using our method, we could detect circulating tumor cells in 86% of patients before treatment of the primary lesion, and the amount of CTCs isolated did not correlate with tumor size or anatomical locations. There was, however, a trend, so this was not significant. The numbers are quite small because most people do not get a biopsy, but there was a trend in increasing circulating tumor cell level and high-risk disease, and this was quantified by MLPA, so somatic copy number alterations. And this led to one of the crucial findings of the study, in that if a patient at this time has 3 or more CTCs, they are more likely to have a significantly worse overall and progression-free survival. And this is simply just by enumerating circulating tumor cells. So above enumerating, I think the field is generally moving towards a more genomics-based approach, so assessing the genetic content within each cell individually. So a couple of years ago, we published showing that the profile of circulating tumor cells not only are remarkably similar to one another, as you can see on this graph here, CDC1 and CDC2, but they also remain relatively stable compared to the primary lesion, as you can see here. So the profiles within these are very similar indicating that they could be used to detect copy number alterations associated with prognosis. So that leads me to some very early preliminary data that is, this is still ongoing work, but these are two examples of circulating tumor cells from primary patients. So if we look at this top figure here, we can see that this patient has a loss of chromosome 3 and no change to 8, meaning that if we go by the TCGA classification of tumors, this person would have a cluster 3 tumor, so a poor prognosis. If we go by the second, we can see there's a gain of 8, which would be a cluster 2 or intermediate prognosis. And the reason this might be useful in the future, pending validation, is that most patients don't have an intraocular biopsy at the time of their primary treatment. And so a lot of people have no idea what their prognosis is. So if we can use a minimally invasive method using circulating tumor cells, as the cell itself is a genomic representation of the disease, we might be able to improve prognostication for patients. Moving on from circulating tumor cells, circulating tumor DNA has really exploded in the last decade or two. There's an enormous amount of work into circulating tumor DNA, and I think a part of the reason for that is it's actually much simpler to implement than circulating tumor cells. Circulating tumor cells can be a bit finicky. And circulating tumor DNA, as I said before, is released from the death of tumor cells. So the apoptosis and necrosis of these cells, it spills the contents into the blood, and you can use, digital PCR or next generation sequencing type methods to detect cancer-specific mutations, and that's how you detect circulating tumor DNA. So quite surprisingly, there isn't actually that much research in circulating tumor DNA and uveal melanoma. So in primary disease, there's still a bit of contention on the detection rate. There's a bit more evidence that it's quite rare and very difficult to see, but there's some evidence that increasing levels are increasing with malignancy. Most of the research has gone into the metastatic disease, and this is because at the metastatic time point, ctDNA is much more detectable. And what all of the studies have shown is that the levels of circulating tumor DNA are associated with poor outcomes and high disease burden. So the more ctDNA have, the worse your outcomes are. And we have summarized most of the literature in our recently published paper. So a couple of years back, a part of the study we published was comparing ctDNA in primary disease versus metastatic. And as you can see from the graph, the levels of circulating tumor DNA at the primary stage are very low compared to metastatic, and the number of patients that have positivity are also very low. Now, because we've been sitting on this data for about 5 years now, you wanted to look at whether or not these levels were associated with any survival differences or prognosis. So we found that there seemed to be a marginally higher hazard ratio for patients with higher ctDNA at this time point, but it was not significant. So it might be worth investigating in the future. But another thing that popped up was that ctDNA in some patients was appearing before radiological progression. So if we take this scan, for example, so we can see NAD, so no active disease, at this time point, there was a very small level of circulating tumor DNA. And this is the concordant PET scan, so you can see there's no disease in this liver. Now, about 6 months later, radiological scans showed there was a small lesion within the liver and circulating tumor DNA rose. So we thought that we would be good to longitudinally monitor patients to see if ctDNA could be used to detect metastases, early or at the same time as radiological scans. So following this up, we found that we could detect circulating tumor DNA at or before this clinical progression in about 63% of patients. And this gave us a sensitivity of 0.54 and a specificity of 1. We also found that, and this is a well-known phenomenon in ctDNA research, but it's always good to see it repeated, is that the levels of circulating tumor DNA at this time point correlate very strongly with the amount of total disease burden, and this is quantified by PET-CT. So on the y-axis, ctDNA, x-axis, disease burden, and you can see there's a very strong correlation between these two. So this led into this survival data. So patients with a high level of circulating tumor DNA. So again, this circulating tumor DNA level is a representation of disease burden. So people at diagnosis with high ctDNA do significantly worse, as you can see on this Kaplan-Meier, with a hazard ratio of 4.64. So it's a good prognostic marker at this time point. Lastly, we wanted to assess, and this is in a small number of patients, undergoing combination ipi-nivo or anti-PD-1. So combination ipi-nivo on the left, anti-PD-1 on the right. So what you can see is that in the patients undergoing combination ipi-nivo, we see a 1 to 2 log reduction in levels. So again, ctDNA is highly correlated with disease burden. So that massive drop at this time shows that there is some response. And this is in line with literature showing that there's some response from ipi-nivo. We don't see that in the anti-PD-1 therapy. And again, this is not a surprise because anti-PD-1 is generally ineffective for treating metastatic uveal melanoma. And this has been shown in a recent abstract published. So this was a conference abstract for ESMO, and it showed that complete clearance of ctDNA upon treatment with tebentafusp, patients who had this had a significantly better overall survival. Again, likely because you have that reduction in disease burden. So I think the evidence currently suggests that for prognostication at primary disease, circulating tumor cells might be a suitable candidate. But for monitoring for disease recurrence, or onset of metastases, using circulating tumor DNA might be useful to intertwine between radiological scans. So in some patients, you might detect it early on. And then for monitoring through treatment, so for example, through tebentafusp, using ctDNA to track disease burden will be able to assist in monitoring how well that treatment is working. So thank you for listening to my talk. I would like to thank all of our study participants who, without their very kind donations, none of this work is possible, as well as my mentors and collaborators listed. Thank you very much.
Great, thanks so much, Aaron. We'll just move straight into questions because there are a few that are appearing. Maybe we'll just stay on this theme of circulating DNA at the moment. So there's a question from Svetlana about the clinical utility of the use of circulating DNA to monitor disease progression or response to therapy. How far away, Aaron, and maybe Sarah might comment on this as well, are we from the sort of level of reliability of this approach, even towards something like NATA accreditation, for it to be truly applicable in the clinical setting?
I think there's some so some ctDNA tests, like for example T790M, have been validated for treatment uses. So there is evidence that circulating tumor DNA can be used for that purpose. I have not sure, however, how long it would take for uveal melanoma to be accredited in that manner, though.
What about things like the commercial, some of the commercial gene panels? Sarah, are you involved in this, in developing such sort of diagnostic level quality assays in your centre?
No, I'm not, but I do know that Boris Bastian in San Francisco, he was using I think, I'm pretty sure, an accredited panel to look at circulating uveal melanoma cells, I mean, and other cancer types. There are panels that have been developed and introduced into the NHS here, but they are predominantly for metastatic carcinomas. But I don't think it'll be long before the melanomas, including uveal melanomas, will be included in these panels and will be accredited. It's just, unfortunately, as I mentioned before, there's a lot of paperwork to go through before you can actually introduce it into the health system.
Yes, so I work within health systems. I know exactly what you're referring to.
I'm wondering, Daniel, if Mark might have phrased
A bit more expansively about the biology of exosomes. Are they released from all cells equally? Are there some cells that release them more than others, et cetera, et cetera?
Sorry, I cut out a bit here, so I missed a bit of the question. But so the exosomes vary in size, but they the ones that we cultured, or sorry, the ones that we examined using nanolight was they were approximately 50 nanometers in diameter. And we had to also confirm that they were indeed exosomes using an immunohistochemical panel. I think there was a question, can you see them using light microscopy? With great difficulty, to be honest. And there was variation between the cell lines and also indeed between the uveal melanoma samples that we obtained from the patients. And it tended to be, but not always, that the more fast-growing tumors, i.e. those which were monosomy 3 or BAP1 mutant, produced more exosomes. But this wasn't statistically significant, but however, there was a trend definitely there. I don't know whether I've answered the complete question.
No, that's great, thank you. So a couple of questions for Peter as well, just probably quite quick ones. Peter, what's the again from Svetlana, what's the percentage of what percentage of uveal melanomas have somatic MBD4 mutations? I think it might have been in one of your slides, as I recall.
Oh, it's a small number. We haven't seen any in our cohorts, but the French group had a couple in their latest paper. So I would guess below 5%.
I think it's about 3 to 4%.
Yeah,
it's really quite low.
We've seen some missenses, but I don't think they are functional. So yeah.
I mean, based on these, very appealing anecdotal case reports relating to this gene, are you routinely sequencing for this for these mutations now, Peter or Sarah, particularly in a diagnostic context?
We mostly do whole exome sequencing, so yeah, then it's just included.
So we aren't at the moment, but it is included in this panel that we're in the process of validating, and so hoping to be able to pick up on a regular basis going forward this mutation in the patients.
Great, and just a final question, also from Svetlana, Peter, regarding potential associations between germline BRCA2 mutations and the development of uveal melanoma. Are you aware of any? Were there any clues about such links in your cohort?
We had a thorough look at our whole cohort 4 or 5 years ago, and we couldn't find any convincing cases at all in our familial data. We had one germline mutation in one case, but it lost the mutant allele in the tumor, so that was not convincing at all. yeah, I'm not a strong believer of BRCA2 in uveal melanoma. I mean, it might
Great.
be, but it's very weak if there's any association, I think.
Great. Well, thanks very much for the speakers for an excellent session. We'll just move on to our last short session of the summit, and that's a presentation by Dr. Tim Luckett. Tim's the co-chair of the Palliative Care Clinical Studies Collaborative and also the co-executive of the Cancer Quality of Life Expert Service Team called Sequest, which is one of the technical services offered by Cancer Australia to the 14 national cooperative cancer trial groups around the country. An incredibly useful resource provided by Cancer Australia for our field with some really excellent people there, and one of which is, of course, Tim. He's also an academic editor for the journal PLOS One, and is going to speak to us briefly. Really, just put I think an open question to our field for potential participation in the development of quality of life tools for patients affected by metastatic uveal melanoma. Thanks very much, Tim, for your presentation, which I'll throw to Dan to close out the session.
Thanks very much, Mark. And I should also, of course, highlight that Professor Anthony Joshua prompted this study by approaching us and suggesting that we embark on the collaboration. And it has also been presented to the AOMA advisory group back in March, who approved it to go ahead. This is our HREC approved recruitment slide. We are inviting anybody in the audience who's living with metastatic uveal melanoma, or any clinicians who are involved in caring for people in that group, to take part in a 30-minute focus group or a one-to-one interview if they prefer, to help us really develop a dedicated quality of life questionnaire for people in this patient group. And I'm hoping that a Zoom poll is up on your screens. And if you think you might be interested in participating, please do enter your email address into that Zoom poll, and I'll send you a bit more information. So just very, very quickly, some background. I don't need to tell this audience much of what I've got up on this screen. The poor prognosis and toxicity of treatments that we've been hearing about today make quality of life a key focus for clinical trials alongside survival, but there is no currently dedicated questionnaire available. Trials to date that have measured quality of life in people with liver metastases have tended to use quality of life questionnaires designed for liver cancer, or those for melanoma, or just for cancer more generally, or indeed the general population. And there are very few trials that have looked at quality of life in people with metastases to sites other than the liver. And there's almost no descriptive data from survey studies or qualitative studies on the experience of living with uveal, metastatic uveal melanoma and its treatment either. So the quality of life questionnaire is currently available for melanoma. There is one under development for metastatic cutaneous melanoma, and we've been in touch with the EORTC team that's developing that, and they agree that probably the issues will going to be very, very different for metastatic uveal melanoma. There is the OP30 for uveal, the MOOD for ocular, and the COMS-QOL for choroidal. But all of these were really developed for people with early stage disease, and they have not been validated in people with metastatic. And then, of course, there are a range of measures from the EORTC and the FACET suites of quality of life measure available either for primary liver cancer or for liver metastases. So really the issue is that the available quality of life questionnaires, they weren't validated for this patient group, and they probably include scales and items that may well be redundant, like vision impairment, vision disturbance and ocular irritation, at least for people who have had their affected eye removed. And of course, these questionnaires may also miss very specific issues that are of concern to people with metastatic uveal melanoma and its treatment. So really, we want to embark on some what's called content validation to see whether we may be able to piece together a questionnaire from existing scales and items from the questionnaires I've just mentioned and any other questionnaires that are currently available, or if we actually need to supplement those with some new items to address any gaps there may be. So that brings us to why I'm here today. It's going to be a qualitative study, face-to-face or Zoom focus groups, depending if we can get a critical mass in one place, we'll do it face-to-face. Otherwise, it can work quite well over Zoom, or one-to-one interviews, particularly for people who are living with metastatic uveal melanoma and would prefer to discuss this in a more private setting. I'm very happy to interview them on a one-to-one basis. Clinicians from any discipline, medical, nursing, allied health, we'd love to hear from you. You all have differing perspectives on the quality of life impacts, and we want as comprehensive an idea as possible. And the analysis will both really be inductive, so we'll be listening to what you say first, but then we will be mapping to these existing quality of life questionnaires to see the degree to which the issues that people are raised are really covered. And then the idea is that we will then develop a new questionnaire that could be subjected to future psychometric testing. And that's quite a rigorous and lengthy process of validation. So just back to my slide again. Just to repeat, 30-minute focus group or interview, and please do put your email into the Zoom poll if you're interested, or of course feel free to send me an email as well at the email address there on the screen. Thanks very much.
Thanks very much, Tim. I hope you get a lot of people joining for that valuable work. Well, this brings us to the end of the 2022 AOMA Summit. So on behalf of the organising committee, sincere thanks to our generous and accomplished speakers, so many of whom have given up their Friday night or Saturday morning sleep to join us. It's really great to have Paul and Jessica's consumer perspective earlier, and also I suspect consumer or patient participation will become a lot more routine at meetings like this in future. On that note, Anthony Joshua has asked me to convey his ongoing appreciation for the efforts of our colleagues at AOMA, the consumer colleagues at AOMA, Susan Vine and others more generally. The sponsors have been very generous and understanding in supporting this summit, particularly given the ongoing uncertainty with the on-again, off-again virtual in-person format. And today certainly wouldn't have been possible without their support. Our sponsors have been Bristol Myers Squibb, Immunocore, Novartis, Idea Biosciences, Eckert and Ziegler, University of Sydney SafeSite Institute, and Peter MacCallum Cancer Centre. Thanks again. Also thanks to Regan Hildebrand-Wise and the team at ASN for hosting this year's summit and ensuring everything's run so smoothly. Also to Maren Morrison, Gabby Byers, Liz Sadowy and their colleagues at Melanoma and Skin Cancer Trials. Dr. Damien Key, whom we heard from earlier, has worked tirelessly in very challenging circumstances to keep us all on track with arranging everything. Thanks, Damien. Finally, thanks to everyone remaining online for your attention and participation throughout the day. We hope to see you all next year. Please help us to promote next year's summit to all your friends and colleagues through whatever means you have at your disposal. Thanks again and enjoy the weekend. Bye-bye.